You know, usually when we talk about a medical diagnosis, there's this expectation of just absolute clinical precision.
Right, yeah.
Like, think about engineering or human medicine. You break your arm, they take an X-ray, it shows that jagged white line on the dark film, and the doctor just points to it, says, "Yep, there it is. There's the problem."
It's binary. I mean, you were looking at a system that allows for really clear, visible categorization. It's either broken or not broken, infected or clear.
Exactly. But then you step out of the sterile hospital and you look at, well, the world of global agriculture. Specifically, if you look at the global citrus industry right now, suddenly that X-ray machine is just completely broken.
Oh, absolutely.
We are looking at a diagnostic landscape that is honestly incredibly murky. I mean, we are dealing with an existential threat to the oranges, the lemons, and the limes that are probably sitting on the counter in your kitchen right now. And the scariest part is just how invisible the threat can be until it's way, way too late.
It truly is the definition of diagnostic muddy waters.
Yeah.
Because the threats we are looking at in commercial citrus, they're microscopic.
Right.
They are highly systemic, and they are evolving and adapting faster than traditional farming methods can even hope to handle. It's basically a biological arms race playing out in slow motion across millions of acres.
Which brings us to the mission of today's deep dive. We are going to explore how an incredibly bizarre caviar-like fruit native to the dense Australian rainforest is somehow, and this is wild, bridging the gap between snowy, freezing greenhouses in Nebraska, high-end metropolitan cocktail bars, and the literal boots-on-the-ground fight to save the global citrus industry from total economic and biological collapse.
The geographic and scientific journey of this single plant is just staggering. I mean, it touches on almost every discipline of modern agricultural science.
Yeah, and to figure out how this is even possible, we've had to pull together puzzle pieces from all over the place for you today. So I've been reading through this surprisingly intense, highly technical Reddit debate on geothermal gardening.
Which is a goldmine, honestly.
It really is. And then trying to square those, like, backyard theories with a deeply detailed entomology study on pest dynamics following a massive freeze.
Right.
Then we're going to dive into a very dense molecular level study from Frontiers in Plant Science. It details citrus genetics. We're also looking at botanical profiles from gardenia.net and some hard-nosed market analysis from the Packer and Central Florida Ag News, just to see how this incredibly complex biology actually translates to the economy.
Yeah, we are connecting dots that most people would never think to put on the same board. It's a phenomenal cross-section of how grassroots backyard experimentation, hardcore laboratory molecular biology, and shifting culinary trends all sort of intersect to solve a global crisis.
So before we get into the heavy science, I want you to simply imagine a fruit. If you were holding it in your hand, it looks a bit like a stubby little gherkin.
Yeah, kind of like a rough, unappealing little pickle.
Right, exactly. But when you slice it open, the inside literally pops out. It looks and feels in your mouth exactly like caviar. And it tastes like a bright, acidic mix of lime, lemon, and grapefruit all hitting your palate at the exact same time.
It's an experience.
It is. And hidden inside the DNA of this strange little oddity, it might hold the genetic superpower to defeat a disease that is currently wiping entire orange groves off the map worldwide.
Okay, let's unpack this.
We need to start by meeting the plant itself in its natural environment. Botanically, the scientific community knows it as Citrus australasica.
Okay.
But out in the world, most people just call it the Australian finger lime. And to truly grasp why its genetics are so highly prized today, you really have to understand the crucible where it evolved.
Right, the environment.
Because this is absolutely not your typical sun-drenched, manicured Florida orange tree growing in neat, irrigated rows. Not at all. The finger lime is native to the lowland subtropical rainforests and the dry, open woodlands of southeastern Queensland and northeastern New South Wales in Australia. Wait, when you say rainforest, my mind immediately goes to an incredibly dense, highly competitive, intensely humid environment, the kind of place where every plant is fighting tooth and nail just to get a sliver of sunlight.
Yes.
Right. Oh, wait. I mean, that sounds like a brutal place for a citrus tree to establish itself.
It is an intensely brutal environment. It grows primarily as a dense understory shrub. This means it's living in the shadows of massive canopy trees, fighting for limited resources.
Wow.
In modern cultivation, farmers usually prune it to keep it between 6 to 12 feet tall for easy harvesting. But out in the wild, left to its own devices, it can scramble and reach up to 33 feet high.
Just trying to push through the canopy to find light?
Exactly. And it is heavily armored. It defends itself with incredibly sharp, stiff 1-inch spines along the leaf axils.
Wow.
Yeah. It has these small, glossy, light green leaves to maximize whatever dappled sunlight it catches. And it produces highly fragrant, white to pink flowers that boast 20 to 25 stamens. It is a rugged, resilient, hardened survivor of a plant.
And then there is the fruit itself, which just feels like pure culinary magic.
Mm.
Think about regular citrus for a second. If you peel a lemon or a sweet orange, you find those little teardrop-shaped juice sacs neatly packed inside the wedge segments.
Right.
And they're all fused together. If you try to pull an orange segment apart with your fingers, it just turns to mush. The sacs rupture and juice gets everywhere.
Yeah, the botanical term for those juice sacs is vesicles. And yes, in traditional commercial citrus, those vesicles are tightly fused.
But the finger lime is completely different. The fruit itself is a small cylinder, usually just 2 to 4 inches long. The rind is rough, pebbled, and it comes in this crazy, vibrant rainbow of colors depending on the specific wild variety. So you get bright green, deep yellow, pale pink, vibrant crimson red, and even a dark purple that looks almost black in the shade.
It's beautiful.
But the real show is what happens when you cut it open. The juice vesicles inside are perfectly spherical, and they remain completely separate from one another. They do not fuse.
Right. They look exactly like tiny, translucent pearls packed tightly inside the rind.
Hence the popular culinary nickname, citrus caviar.
Exactly.
And because those pearls are separate and have a firm outer skin, they don't turn to mush. When you eat them, they literally burst on your tongue when you bite down. It is a massive textural explosion.
It really is.
But I do have a question here, or at least a conceptual hurdle I'm struggling to wrap my head around based on our sources. If this plant is native to a subtropical Australian rainforest, growing in the humid shade of a canopy, how on earth is it popping up in everyday conversation for backyard gardeners in completely different, wildly unsuitable climates?
Yeah, it's a fair question.
I mean, you see people growing them all over the place now.
Well, that adaptability is the true beauty of its genetics. What's fascinating here is that despite its exotic, very specific rainforest origins, the finger lime is remarkably plastic in its environmental tolerance.
Plastic meaning flexible.
Yes. It thrives across USDA hardiness zones 9 through 11. It prefers well-draining, slightly acidic soil, but it is exceptionally tough once its root system is established. And we aren't the first people to realize how valuable it is.
No, really?
No, human beings have recognized its utility for a very long time. Aboriginal Australians have utilized the finger lime for millennia. They deeply integrated it into their survival and culture.
I am going to assume they weren't using it as a fancy garnish for their drinks.
Far from it. During harsh, dry seasons, the fruit was a crucial source of vital hydration and dense nutrition when water was scarce.
That makes sense.
But it was also highly valued medicinally. They used the juice, and sometimes the crushed skin, to treat cuts, minor wounds, and skin infections.
Wait, really? Like an antiseptic?
Yeah. And when we apply modern scientific analysis to its nutritional profile today, that ancient medicinal application makes perfect logical sense.
Is that tied to the vitamin C content? Because citrus is always famous for that.
Vitamin C is a major component, yes. A standard 100-gram serving of finger lime provides between 80 to 88 milligrams of vitamin C.
Wow.
To put that in perspective, that is nearly 98% of the daily recommended intake for a human adult. But it goes far beyond just vitamin C. Combine that with an incredibly dense concentration of powerful antioxidants.
Like what kind?
Specifically phenolic compounds and flavonoids. So you have a fruit that naturally combats cellular oxidative stress and topical inflammation. What's fascinating here is how this historical evolutionary survival mechanism, both for the plant defending itself in a hostile rainforest and for the humans utilizing it for medicine, translates directly into modern agricultural resilience.
So the exact same chemistry that helped heal wounds thousands of years ago is what scientists are looking at today.
Exactly.
Okay, so we know this plant has evolved incredible defensive armor and chemical resilience in the Australian heat. But the obvious question is, how far can you push that resilience? If you take it out of the rainforest, does the armor hold up against the cold?
That's the big test.
Right. And that actually brings us to a wildly fascinating, almost absurd experiment happening in, of all places, the snowdrifts of Nebraska. Because nothing says subtropical rainforest fruit quite like the freezing plains of the American Midwest.
Quite the visual.
We pulled this amazing thread from the slash gardening subreddit. It focuses on a retiree living in Alliance, Nebraska, and he is successfully growing citrus in the dead of winter using a geothermal greenhouse.
It is a brilliant, highly practical example of extreme cultivation and applied human ingenuity.
Oh, totally. The Reddit debate on how this gardener actually pulls this off is so entertaining, but is also an incredible lesson in thermodynamics. The basic premise is that he is tapping into the Earth's ambient underground temperature. So if you dig down a few feet, the Earth stays at a constant 54 degrees Fahrenheit year-round, regardless of the blizzard happening on the surface. He is using a system to pump that 54-degree air into his greenhouse to keep the ambient temperature above 28 degrees Fahrenheit.
And that 28-degree mark is critical
Uh-huh.
because that is the absolute danger zone where citrus trees begin to suffer irreversible tissue damage and freeze to death.
And naturally, the internet gardening community had some very strong opinions on his chosen method of thermodynamic engineering.
Oh, absolutely. They tore into the physics of it.
They really did. To visualize what they were arguing about, think about the physical act of carrying water across a yard. If you try to carry water in a bucket full of holes, you're going to lose a massive amount of it along the way.
Obviously.
If you carry that same water soaked into a dense sponge, you will hold on to it much better. But the sponge itself is incredibly heavy and requires a lot more physical effort to carry. So in the Reddit thread, a user named Washington Jacksons points out that using air to transfer heat is essentially using a leaky bucket.
Right, because air is a notoriously terrible conductor of heat.
Exactly. They noted that if the air coming out of the underground tubes is 54 degrees, but the greenhouse is struggling to stay at 28 degrees, the system is losing 26 degrees of vital heat through the thin walls of the greenhouse. So they argued that the gardener should be using a small water recirculating pump through underground pipes instead of an air blower, because water has a much higher capacity to hold and transfer thermal energy.
It is a perfectly valid critique from a pure physics perspective. They are debating specific heat capacity. Water requires significantly more energy to change its temperature than air does, which means it also retains that heat much longer when exposed to cold environments. It is a vastly superior thermal battery.
Right, makes total sense. But then another user, Cookie-san, jumps into the thread and counters with the energy cost of moving the heavy sponge versus moving the leaky bucket.
Ah, the operational cost.
Yes. They say, sure, water holds heat exponentially better, but pushing heavy liquid water through hundreds of feet of underground piping requires a significantly larger, much more energy-intensive mechanical pump than simply blowing air through a wide tube with a fan.
Right, moving air takes vastly less electricity.
So Cookie-san argues that the air system, while less efficient at thermal transfer, is actually far more efficient from a financial and energy consumption standpoint for a backyard hobbyist.
It is a classic engineering trade-off. Capital costs and operating costs versus raw thermal efficiency. But if we connect this localized backyard debate to the bigger picture of the global citrus industry, this Nebraska experiment visually proves a fundamental biological rule.
Which is?
Citrus can technically survive extreme freezing conditions,
Right.
but it requires massive, resource-intensive human intervention and completely enclosed, artificially heated microclimates.
And you simply cannot construct a climate-controlled geothermal glass bubble over 10,000 acres of commercial orange trees in Florida or Brazil. It's economically impossible.
Exactly. Commercial agricultural groves are completely exposed. They are entirely at the mercy of macro weather patterns. And that terrifying vulnerability was brutally exposed very recently.
Yeah.
In December 2022, northern Florida experienced an absolutely devastating historic freeze.
I remember reading the raw meteorological data on this in our source stack. This wasn't just a chilly morning where you put a blanket over your tomatoes. This was catastrophic.
It was a generational freeze event. In the Florida Panhandle, temperatures plummeted down to 17 degrees Fahrenheit, which is roughly negative 8.3 degrees Celsius.
Wow.
And it didn't just dip down to that temperature for a few minutes before sunrise and bounce back up. It stayed locked at that bone-chilling 17 degrees for nearly a full hour.
That's so long for a plant.
It is. Furthermore, the region experienced sustained freezing temperatures for 4 consecutive days.
What actually happens inside the biology of a citrus tree when the air around it is 17 degrees for that long? I mean, structurally.
The physical damage is horrific at a cellular level. Plants are primarily water. When temperatures drop that low for that long, the water inside the plant cells literally freezes and expands. It forms these jagged ice crystals that puncture the cell walls from the inside out.
Oh, wow. It basically shreds the plant from the inside.
Yes, it caused catastrophic structural damage to the groves. Complete defoliation occurred. The trees lost absolutely all their leaves. And in many of the younger, more vulnerable groves, the cellular damage was so extensive it resulted in total tree death.
That's heartbreaking.
It is. The archival pictures from the North Florida Research and Education Center show these haunting, completely brown, withered husks standing in rows where vibrant green trees used to be.
You know, when I first read about that level of deep freeze devastation, my immediate thought was about the pests.
Ah,
Right. Because a freeze that is cold enough to shatter the cellular walls of a tree must absolutely obliterate the insect population living on it, right? I assumed there had to be a silver lining for the farmers. The bugs must have frozen solid, giving the surviving trees a fresh, pest-free start the next spring.
it is a very logical, hopeful assumption.
But I'm wrong.
Well, agricultural biology is rarely that accommodating to our expectations.
I found that out the hard way when looking at the data. We reviewed an incredibly detailed 2-year entomology survey. This was conducted by the Florida entomologists right after that 2022 freeze, spanning through 2023 and 2024.
Right.
And the researchers set up field monitoring to track 4 main citrus pests to see exactly what happened to their population dynamics after the ice thawed. They tracked citrus leaf miners, red scales, ambrosia beetles, and the Asian citrus psyllid. And the results completely shattered my assumption.
The data is genuinely eye-opening, and it highlights how complex these ecosystems are.
Hmm.
The researchers use something called ARMA model forecasting alongside rigorous biweekly field sampling. They monitor populations in 3 different commercial groves and several residential backyard settings to get a comprehensive view.
Wait, before we get to the bugs, what exactly is an ARMA model? How do statisticians use a model to predict what a bug is going to do?
So ARIMA stands for Auto-Regressive Integrated Moving Average.
Okay, that sounds intense.
It does, but without getting bogged down in the mathematics. It is a sophisticated statistical algorithm used to analyze and predict time series data. In entomology, you feed the model years of historical data on bug populations. You factor in seasonal cycles, temperature trends, and previous population spikes.
Sort of teaching it the history.
Exactly. The model then forecasts what the population should look like under normal conditions. By comparing the actual post-freeze bug counts in the field against the ARIMA forecast of what a normal year would look like, the researchers could definitively measure the exact statistical impact of that specific freezing event.
So it's basically a mathematical baseline to show just how weird things got after the ice storm.
Correct.
And the freeze did not wipe the bugs out uniformly at all.
Not even close. The ambrosia beetles and the red scales, which are pests that damage the woody bark and degrade the cosmetic quality of the fruit, they essentially just rode the storm out. Yeah. Their population stayed at relatively low, stable levels, largely unaffected by the deep freeze. Their physical location on the thick bark likely provided enough insulation to survive.
Wow.
But the real shocker, the anomaly in the data, was the citrus leaf miner. Far from being wiped out by the cold, the leaf miner population absolutely exploded in the summer of 2023, just months after the devastating freeze.
Exploded.
The researchers counted over 95,000 leaf miners in their field traps.
Okay, how is that biologically possible? If the host tree is dead or severely damaged and its leaves have all fallen off, how do the tiny bugs that rely on eating those leaves multiply by the tens of thousands?
It comes down to the cascading biological mechanics of how damaged trees attempt to recover, combined with a disruption in predator-prey dynamics.
Okay.
When the extreme cold hit, the surviving trees were thrust into severe physiological stress. To conserve energy and survive, they were forced to drop all their damaged leaves. They were completely bare, standing dormant.
Absolute survival mode, just shutting down non-essential systems.
Yes, but a tree cannot survive without leaves indefinitely. When the weather finally warmed up in the spring and early summer of 2023, those surviving trees desperately needed to restart photosynthesis to generate energy and heal.
Right.
So instead of pushing out leaves gradually over time, they rapidly pushed out a massive, highly synchronized flush of brand new leaf growth all at once across the entire grove.
Oh, I see. It's essentially an all-you-can-eat buffet of the most tender, soft, vulnerable leaves served up simultaneously.
Exactly. An unlimited concentrated food source suddenly appeared right when the surviving leaf miners were emerging. But there is a crucial second factor that created this perfect storm.
What's that?
The extreme frost didn't just affect the pests, it likely decimated the populations of the leaf miners' natural enemies. Specifically, the freeze was lethal to a tiny parasitoid wasp called Ageniaspis citricola, which normally hunts and kills the leaf miner.
Ah. So
Yeah.
the freeze killed the cops, but the robbers managed to survive in the crevices. And when spring came, the bank vault of new leaves was left wide open with zero security.
That is a highly illustrative way to look at it. With an infinite supply of fresh, tender foliage to consume and absolutely zero predatory wasps to keep their breeding cycle in check, the leaf miner population surged to apocalyptic levels, far higher than the researchers observed in the much milder, unfrozen year of 2024.
Here's where it gets really interesting. Because as incredibly destructive as 95,000 leaf miners are, and they do massive damage, tunneling through the leaf tissue, leaving these little silvery winding trails that physically weaken the leaf and open the tree up to secondary bacterial infections like citrus canker, the leaf miner isn't actually the main character in our agricultural horror story today.
No, it's not.
The true villain hiding deep in this entomology data is the fourth bug they tracked, the Asian citrus psyllid.
The psyllid is the true threat. It is the vector. It is the biological delivery mechanism for the most catastrophic disease the global citrus industry has ever faced.
And what's wild to me is that the researchers didn't even find high concentrations of these psyllids in the heavily monitored commercial groves. According to the study, they found the psyllids lurking almost exclusively in residential backyard citrus trees. Someone's forgotten. Unsprayed pet lemon tree sitting behind a fence in Bristol or Apalachicola is quietly harboring the worst enemy the multi-billion-dollar industry has ever faced.
It's sobering.
The bug itself isn't a voracious eater like a locust. It's tiny. So why do we care so much about this one specific insect? Because it operates like a Trojan horse. It carries a disease called HLB.
One long ding. Globally, it is also known as citrus greening disease. The disease is caused by a highly destructive phloem-restricted bacteria named Candidatus liberibacter asiaticus.
That's a mouthful.
It is. So in the scientific literature, we often abbreviate that long name simply to CALIS. And to truly understand the sheer gravity of HLB, we need to zoom in and look at the microscopic molecular battlefield inside the plant itself, which is brilliantly detailed in the Frontiers in Plant Science study.
Okay, let's slow down and dive into that because I need a masterclass on how a microscopic bacteria actually kills a tree that is 20 feet tall. It's not like a caterpillar mechanically chewing through the trunk until it falls over.
No, it is far more insidious and complex than mechanical damage. The process begins when the Asian citrus psyllid lands on a citrus leaf to feed on the plant's sap.
Okay.
As it inserts its mouth parts into the leaf tissue to feed, it inadvertently injects the callus bacteria directly into the plant's phloem.
A phloem, right?
The phloem is the critical vascular system of the tree. Think of it as the tree's circulatory system. It is the living pipeline responsible for transporting the vital sugars and metabolic products created during photosynthesis in the leaves, moving them downward to feed the roots and outward to develop the fruit.
So by injecting it into the phloem, the bug has essentially put the bacteria directly into the tree's bloodstream. It has systemic access immediately.
Precisely. Now, it is important to note that the tree doesn't just sit there passively and let the bacteria multiply. Plants have highly evolved, complex immune systems. Right. The tree's cellular sensors detect the presence of the invading callus bacteria, and the immune system goes into absolute overdrive. It initiates a massive panic defensive response. A key component of this initial immune response is the rapid, overwhelming generation of molecules known as reactive oxygen species, or ROS.
Is that similar to what happens in the human body, like an oxidative burst to kill an infection?
Yes. The tree is actively trying to use these highly reactive, unstable molecules to essentially burn, degrade, or chemically neutralize the invading bacteria. It is a chemical scorched earth tactic.
Wow!
But there is a severe cost. Producing that massive volume of ROS causes intense oxidative stress throughout the entire biological system of the plant. The ROS molecules are so volatile that they begin damaging and degrading the plant's own healthy cellular machinery, breaking down cell membranes and disrupting normal protein function.
So it's like a human body running a fever so high to kill a virus that the heat actually starts causing neurological damage. The defense mechanism itself becomes highly destructive.
That is one crucial part of the physical decline. But the fatal killing blow to the tree comes from how its immune system attempts to physically quarantine the bacteria.
Oh, it tries to trap it.
Yes, to stop the callus bacteria from flowing freely through the vascular system and infecting the roots, the tree's immune system triggers the rapid, heavy deposition of a biological substance called callus.
Callus. The study mentioned this is a type of carbohydrate, right?
Yes, callus is a beta-1-gru-3 glucan polysaccharide. To visualize it, think of callus as a thick, dense molecular spackle or a sticky biological plaque.
Okay.
The tree desperately plasters this callus all along the sieve plates, the microscopic connecting valves of the phloem. It is trying to build a physical wall to seal off the infection site.
Wait, so the tree is essentially having an extreme autoimmune response.
Yes.
It's like a severe allergic reaction where a human throat swells shut to stop an allergen, but it ends up asphyxiating the person. The tree is so desperate to trap the bacteria that it ends up pumping molecular spackle into its own veins and completely choking off its own vascular system.
That analogy is incredibly accurate. The plant effectively strangles its own nutrient supply lines. And the downstream macroscopic effects of that internal choking are what a farmer actually sees when they walk through an infected grove.
Because the sugars can't move.
Exactly. Because the phloem pipeline is blocked with callus, the essential sugars created by the leaves during photosynthesis have nowhere to go. They physically cannot travel down the trunk to feed the root system, and they cannot travel to the branches to plump up the developing fruit.
The pipeline is entirely clogged. The factory is producing food, but the delivery trucks are trapped in the driveway.
Consequently, those trapped sugars just back up and accumulate inside the leaves. This leads to a massive, highly toxic buildup of starch directly inside the chloroplasts, the very cellular engines responsible for photosynthesis.
Right.
The starch granules swell and become so large that they physically rupture and destroy the delicate chloroplast structure, completely halting the leaf's ability to turn sunlight into energy. Visually, to the naked eye, this presents as a classic symptom called blotchy mottle.
What does that look like?
These asymmetrical, sickly yellow patches spreading across the green leaves.
And because the roots below ground and the fruit on the branches are literally starving for those trapped sugars, the tree panics and starts dropping its fruit prematurely, just aborting it to save energy.
Yes, fruit drop is a major sign.
And the meager fruit that does manage to cling to the branches is warped, misshapen, stays green instead of turning orange, and tastes incredibly bitter and salty because it lacks sugar development.
And the tragedy of this biological cascade is that right now, despite hundreds of millions of dollars invested in research, despite experiments with thermotherapy, nutritional supplements, and aggressive chemical treatments, there is no known cure for an infected commercial citrus tree.
Nothing.
Once the callus bacteria is established and the callus response begins, it is a death sentence. The tree will slowly, inevitably starve to death, strangled by the overreaction of its own immune system.
Man. Which brings us all the way back around to our weird, thorny little hero from the dense Australian understory, the finger lime.
Yes. Enter the genetic rescue mission.
Dr. Menjul Dutt and his team of researchers at the University of Florida's Citrus Research and Education Center made an absolutely incredible discovery. The Australian finger lime, along with some of its close wild relatives, possesses a natural, profound genetic tolerance to the callus bacteria.
It is a scientific game changer. These wild plants do not just passively endure the disease, their genetics actively resist the catastrophic physiological breakdown that our commercial oranges and grapefruit suffer.
Wow.
The bacteria might be present, but the finger lime doesn't panic and choke itself to death. But the researchers knew they couldn't just tell Florida farmers to abandon a century of tradition, stop growing juice oranges, and plant thousands of acres of thorny wild shrubs.
Right. People want orange juice, not caviar water.
Exactly. So they needed a way to transfer that ancient genetic superpower into the vulnerable citrus varieties we already rely on.
So they started playing molecular matchmaker. They initiated this incredibly cutting-edge breeding program, attempting to forcefully merge the unyielding, battle-tested genetics of the finger lime with highly vulnerable, commercially valuable citrus varieties. And they didn't just rely on one single method. They started with conventional breeding the old-fashioned way. They physically took pollen from finger limes and crossed them with red pulp pomelos and sweet mandarins, planted the resulting seeds, grew the hybrid trees, and waited to see which ones inherited the best traits.
Which is an excruciatingly slow process. Citrus trees have a notoriously long juvenile period before they mature and bear fruit, and their genetics are incredibly complex and unpredictable.
So to speed things up, they also went full sci-fi. The study details how they used an advanced technique called somatic cell fusion. They bypass flowers and pollen entirely.
Somatic cell fusion is a fascinating piece of biotechnology.
I really need you to break down how that actually works because the study talks about fusing protoplast to create a tetraploid organism in a petri dish, and I feel like I'm reading a science fiction novel.
It sounds like it, yeah.
Yeah.
Let's demystify the terminology. A typical plant cell is surrounded by a rigid, protective outer shell called the cell wall. A protoplast is simply a living plant cell that has had that hard cell wall chemically or enzymatically stripped away, leaving only the delicate inner membrane and the genetic material inside.
Okay, so they strip the armor off the cells to expose the core?
Exactly. In the lab, Dr. Dutt's team took protoplasts from an OLL8 sweet orange, a highly vulnerable commercial juice variety, and they took protoplasts from a resilient finger lime. By applying specific electrical pulses or chemical agents in a petri dish, they forced these two naked cells to physically fuse together into one single massive cell.
They literally mashed the two cells together. What happens to the DNA?
It combines. This creates what is known as a tetraploid organism.
Hmm.
To understand that, remember that normally a plant or a human gets half of its DNA from one parent and half from the other. We call that diploid.
Right.
But in this somatic fusion, the new cell retains the entire genetic instruction manuals of both parent plants. It has 4 sets of chromosomes instead of 2. They successfully crammed the complete genome of a sweet orange and the complete genome of a finger lime into a single living hybrid cell, which they then coaxed into growing into a full tree.
That is mind-blowing. But the goal wasn't to grow a tree that produces a bizarre orange caviar hybrid fruit. The goal here was to create new, superpowered rootstocks.
Yes, the application of this genetics is focused entirely below the soil line.
So what does this all mean for the farmer? If you spend years in a lab creating a superpowered HLB-resistant root system, how does that actually help the normal orange growing on the branches at the top of the tree?
If we connect this specialized lab work to the bigger picture of how commercial citrus is actually grown in the field, almost no commercial citrus tree in the world is grown from a seed on its own natural roots. That is too unpredictable.
Okay.
Instead, the industry relies entirely on grafting. A grower will take the root system of a hardy, soil-adapted plant, this is called the rootstock, and they will physically splice or tape the upper branches of a fruit-bearing plant called the scion, like a Valencia sweet orange, onto the trunk of that rootstock.
I see. So you cut them both, bind the open wounds together, and as they heal, the vascular systems connect and they grow as one single frankentree. The hardy roots pump water up to the delicate orange branches.
Exactly. What Dr. Dutt's team discovered is the true breakthrough of this entire saga. They found that if you take a highly vulnerable Valencia sweet orange scion and you graft it onto one of these newly created HLB-tolerant hybrid rootstocks, like a mandarin finger lime cross, they designated MFL 198, or a pomelo finger lime cross named PFL 261, that hardy rootstock acts like a systemic biological immune upgrade for the vulnerable orange branches living above it.
That is wild. The roots below ground are literally sending chemical defense signals up the trunk into the canopy, teaching the orange leaves how to fight a disease they have no natural immunity to.
Too incredible.
And we have the hard molecular proof of this happening from that massive 24-month greenhouse study. They took these grafted trees, deliberately inspected them with the deadly HLB bacteria using psyllids, and then tracked them continuously for 2 years using incredibly precise quantitative PCR testing to measure the exact bacterial load inside the leaves.
And this is where the raw data gets incredibly compelling. When researchers use qPCR testing to measure bacterial infection, they look at a metric called the C value, or cycle threshold.
Right.
Without diving too deep into laboratory protocols, the most vital concept to grasp is that the C value represents an inverse relationship to the amount of bacteria present.
Explain how that inverse relationship works.
The PCR machine works by repeatedly heating and cooling the sample to amplify or copy the target bacterial DNA until there is enough of it for the machine's sensors to detect. Each round of heating and cooling is one cycle.
Okay.
If a plant is heavily infected and absolutely swarming with bacteria, the machine doesn't have to run very many cycles before it detects the DNA. It finds it quickly. Therefore, a lower C8 value means there is a higher, much more severe amount of bacteria present.
Oh, I get it.
Conversely, if the plant is relatively healthy and has very little bacteria, the machine has to run cycle after cycle after cycle, amplifying the tiny amount of DNA until it finally registers. So a higher seed value means there's significantly less bacteria.
Got it. Low number equals a very sick tree. High number equals a healthy tree. That makes perfect sense.
Precisely. So let's look at the control group. They tested vulnerable Valencia oranges grafted onto standard swingle rootstocks. which is the most common commercial rootstock used in the industry right now. After 24 months of deliberate HLB infection, those control trees returned a seed value of 22.25.
And that's bad.
In the world of citrus pathology, a 22 indicates a highly active, severe, unmanageable bacterial infection. Those trees were dying.
But what happened when they tested the Valencia oranges that were grafted onto the experimental finger lime hybrid roots?
The difference was staggering. Their seed values were significantly higher across the board. Some of the specific hybrid combinations pushed the seed values all the way up to 35.00.
Wow.
At a seed value of 35, the bacterial titer is so incredibly low that the infection is barely registering on the equipment. The bacteria is present, but is completely suppressed. And even more astounding, when the researchers tested the tissue of the hybrid rootstocks themselves, below the graph line, those roots tested completely negative for callus.
Wait, completely negative?
Yes, the bacteria was circulating in the top of the tree, but it couldn't even establish a foothold or survive in the hybrid root system.
Okay, so the hybrid roots are essentially immune, and they are somehow projecting a protective chemical shield upward into the vulnerable orange leaves. How on earth are they doing that? What is the actual biochemical armor they are manufacturing and sending up to the sweet orange?
The Frontiers study broke this mechanism down beautifully through metabolic profiling. The hybrid rootstocks were fundamentally altering the biochemistry of the Valencia cyan attached to them. First, they observed that the trees grafted on hybrid roots were able to maintain much higher, stable levels of chlorophyll even while under infection.
Oh, so they stay green.
Exactly. While the control trees on standard rootstocks quickly turned yellow and lost their ability to photosynthesize, the hybrid grafted trees stayed vibrant green and metabolically energetic.
They managed to keep their cellular solar panels running despite the infection trying to shut them down.
Precisely. Second, the hybrid roots somehow helped the orange canopy manage that toxic starch accumulation we talked about earlier. They prevented the chloroplasts from choking to death on backed up sugars, keeping the vascular flow open.
Which means the tree didn't develop that sickly, blotchy model, and it didn't starve its own fruit.
Correct. But the real heavy lifting, the core of the defense, was driven by secondary metabolites. The researchers conducted chemical assays and found massive, sustained spikes in total phenolic compounds, or TPC, and total flavonoid content, TFC, directly inside the vulnerable leaves of the hybrid grafted trees.
Wait a minute. Phenolics and flavonoids, those are the exact same antioxidants we were talking about earlier. The ones that made the wild finger lime so valuable in ancient Aboriginal medicine, the compounds that promote human healing.
It is the exact same chemical family, yes. Phenolics and flavonoids are incredibly potent, naturally occurring antioxidants. In biology, they act as powerful free radical scavengers.
Right.
Think back to the oxidative burst. When the tree's immune system detects the HLB bacteria and triggers that massive panicked production of dangerous reactive oxygen species, these flavonoids rush into the cellular fluid and chemically neutralize the volatile ROS molecules.
Wow.
They act as a chemical fire extinguisher, putting out the internal inflammatory fire before the ROS can damage the tree's own healthy cell walls.
That is an incredibly elegant biological system. And it's not just a passive chemical shield either. The study actually tracked the real-time genetic expression happening inside the orange leaves. They proved that the hybrid rootstocks were actively causing the Valencia leaves to highly upregulate to turn on specific targeted defense genes. They highlighted one gene in particular called beta amylase 6.
Yes, beta amylase 6 is an enzyme gene directly responsible for starch degradation. When this gene is upregulated, the plant produces enzymes that rapidly break down complex starches into simple, movable sugars, ensuring they don't crystallize and clog the chloroplasts.
They also recorded massive spikes in the expression of genes like glutathione S transferases and thioredoxin reductase. That sounds like a mouthful of jargon. What are those genes actually doing inside the leaf? Are they acting like cellular janitors?
Cellular janitors is a perfect way to conceptualize it. Those specific gene families are critical for deep cellular detoxification and for maintaining the delicate redox balance inside the cell.
Okay.
When the cell is under stress and filling up with toxic byproducts from the bacterial infection and the immune response, these upregulated genes produce enzymes that bind to the toxins, neutralize them, and sweep them out of the functional areas of the cell. Essentially, the hybrid rootstock is sending a chemical survival manual up the trunk, actively teaching the vulnerable orange tree how to clear out the toxic debris of the infection and survive the onslaught. It is an absolute masterclass in plant resilience.
It really is. It's breathtaking science. But as we pivot to the final chapter of this story, we have to pull our heads out of the microscope and look at the harsh reality on the ground. Because saving the molecular biology of the orange tree in a pristine university greenhouse is one thing. Saving the livelihood of the multi-generational citrus farmer who is facing bankruptcy is another entirely.
That's the real challenge.
And this is where we turn to the economic analysis from the packer, which dives into the fascinating reality of actually selling this caviar.
It is a vital perspective.
Yeah.
Because agricultural science does not happen in a vacuum. It happens in a living, breathing, often volatile economy.
Right. And there is this beautiful, entirely unintended market consequence that unfolded while Dr. Dutt was locked in his lab trying to save the commercial juice orange. According to the reporting, a local citrus grower approached Dr. Dutt and basically said, "Hey, it's incredible that you're making these complex tetraploida hybrids to save my orange rootstocks. But do you have any idea how much money fancy chefs in the city are paying for the actual pure finger lime fruit itself?"
The culinary demand was exploding, completely independent of the agricultural crisis. High-end restaurants were begging distributors for supply.
Yeah, the grower told Dr. Dutt he had just come back from a trip to California and paid an absolute fortune for a single upscale dish that featured just a few finger lime pearls as a garnish. The culinary world, avant-garde mixologists, high-end craft bartenders, Michelin-starred chefs, they desperately want the caviar.
Right.
So Dr. Dutt realizes he is sitting on a brilliant two-for-one opportunity. He can continue using the wild genetics to breed resistant rootstocks, but he can simultaneously breed specialized, pure finger lime varieties for Florida growers to plant and sell right now as a highly lucrative cash crop.
Which is an economic lifeline that is desperately needed in rural Florida, given how many thousands of acres of traditional groves have been entirely destroyed and abandoned due to the HLB epidemic.
So adapting to the market, the university breeding program officially releases 2 brand new, specifically Florida-adapted finger lime cultivars for commercial growers. The first is called UF Sunlime. It produces a relatively large cylindrical fruit, about 2.7 to 3.1 inches long, and crucially, it retains that inherent HLB tolerance.
Nice.
The second cultivar is UF Red Lime, which is much longer, reaching 4.2 to 4.8 inches, and is rated as highly HLB tolerant. And my absolute favorite fun fact from this entire deep dive, the scientists explicitly bred these new commercial cultivars to produce pearls that are deep red and pink inside rather than the traditional pale green.
Oh, really?
Yeah. Why? Because Dr. Dutt conducted an informal consumer survey, and 9 out of 10 people explicitly said they preferred the visual pop of the red pearls over the green ones.
It is a brilliant, highly pragmatic intersection of hardcore agricultural resilience and basic consumer psychology. In the high-end culinary world, people eat with their eyes first. The visual appeal drives the premium price.
And we have a tangible, real-world example of this economic pivot playing out right now. We read the profile of Brian Wilder, a veteran citrus grower featured in both the Packer and Central Florida Ag News. This man's story represents the entire industry. He owned and operated a traditional navel orange grove in Plant City, Florida. But by 2016, due to the total unmanageable collapse of his trees from citrus greening, he was forced to shut the entire operation down.
That's rough.
He abandoned his family grove and sat on the sidelines for years watching the industry crumble.
It is a tragically common narrative across Florida. Generational farmers forced to walk away from their land because the biology simply wouldn't support the economics anymore.
But then, in the summer of 2022, Wilder attends an agricultural seminar given by Dr. Dutt. He listens to the presentation about the finger limes, he hears the hard data about their inherent genetic resistance to greening, and he hears the market reports about the soaring culinary demand from restaurants. And he decides to take a massive risk and jump back into the industry. In August of 2024, he planted 900 new finger lime trees on his dormant family farm.
And how are those new plantings performing in the field compared to his old navel oranges?
Unbelievably well. He reported that cultivating them is fundamentally similar to managing any other citrus crop. They require the same basic herbicide regimens, the same irrigation setups, the same foundational care.
That's good news.
But remember that brutal, historic 17-degree Florida freeze we spent so much time analyzing earlier? The one that caused total defoliation and death in regular citrus groves? Wilder noted that his young finger limes went through a severe deep freeze in January shortly after planting, and they didn't suffer a single spot of leaf burn. Their wild, resilient genetics held up perfectly against the cold shock.
So the field agronomy is solid. The plants can demonstrably survive both bacterial disease pressure and the extreme weather events. But what about the back end? What about the economics of actually moving the product?
And that is the massive looming hurdle that could derail the entire rescue mission. We hear from Angelia Chen, an agricultural economist at the University of Florida, and she lays out the stark financial reality. The biggest challenge for these farmers isn't growing the finger limes, it is successfully selling them at scale.
Right.
Right now, citrus caviar is strictly a luxury specialty product. It commands exorbitantly high prices per pound precisely because there is very little acreage planted. It thrives on scarcity. It's a niche novelty for the wealthy.
And the average everyday consumer walking through a standard grocery store has absolutely no idea what a finger lime is, how to cut it, or what to do with the pearls in their home kitchen.
Right. You cannot just dump a massive wooden bin of spiky, dark, rough, finger-shaped fruits next to the familiar, friendly, bright yellow lemons and expect suburban families to toss them into their shopping carts for Tuesday night dinners. There is zero consumer education. So, the university economists and the pioneer growers are having to build an entirely new, highly specific marketing strategy from scratch.
They have to target the trendsetters to build cultural cachet.
Yes, they are heavily targeting craft mixologists. Imagine sitting at an upscale bar and ordering a spicy michelada or a premium gin and tonic. Instead of a boring standard lime wedge perched on the rim, the bartender spoons in a cluster of these vibrant ruby red pearls.
That sounds amazing.
The pearls float suspended in the carbonation, and they deliver a sharp textural burst of concentrated lime flavor only when you sip and bite down. They are targeting high-end, innovative chefs, convincing them to use the caviar as an interactive, textural garnish on delicate dishes like scalloped ceviche or fresh oysters, instead of just indiscriminately squirting generic acidic lime juice over the whole plate and ruining the texture.
It completely elevates the sensory dynamics of the dining experience, which justifies the premium price point the farmers need to survive.
And in the modern era, they are heavily relying on the visual power of social media influencers. Platforms like TikTok and Instagram are the perfect vehicle for this specific crop. The finger lime is an inherently dramatically visual ingredient.
Oh, for sure.
Watching a macro lens video of someone slicing open a red finger lime and squeezing the rind, seeing that cascade of crystalline caviar erupt onto the cutting board, is incredibly satisfying to watch on a smartphone screen. It is tailor-made for viral food trends.
This marketing push raises an incredibly important, dangerous question, though, and it's one that economist Angelia Chen specifically warns the industry about. How do you rapidly scale up the production of a niche, luxury crop without accidentally crashing your own fragile market?
You mean falling into the classic boom-bust agricultural cycle?
Exactly. Right now, the profit margins look incredibly attractive because the supply is highly restricted. But if every struggling, desperate citrus grower in Florida reads these reports and suddenly decides to plant 1,000 finger lime trees tomorrow, the wholesale market will be completely flooded with fruit in 3 to 4 years when those trees mature.
Oh, I see.
If consumer demand and everyday culinary awareness haven't grown at the exact same exponential rate to match that massive influx of new supply, the price per pound will plummet overnight, and these farmers will go bankrupt all over again, this time with healthy trees.
It is a terrifyingly delicate economic tightrope they have to walk.
It requires immense discipline. It requires slow, steady, meticulously data-backed supply chain development. The industry has to actively build the consumer appetite at the exact same pace they are putting trees in the ground.
Like prepping the market.
Yes. They have to invest heavily in public education, funding in-store sampling stations, engaging with local farmers markets, sponsoring those viral cooking videos so that when the large commercial-scale harvests finally arrive in a few years, there's already an eager, educated consumer base waiting to buy them.
It really is a masterclass in agricultural economics happening in real time. So to bring all these disparate threads together and look at the whole tapestry, think about the sheer improbable journey we've just tracked over the last hour. We started by looking at a wild, thorny, unassuming shrub hiding in the dense, competitive understory of the Australian rainforest, a plant utilized for millennia in ancient Aboriginal medicine to heal human wounds with its dense concentration of antioxidants.
A seemingly ordinary plant shaped by millions of years in a harsh, unforgiving environment into an impenetrable genetic fortress.
We watched that biological fortress get rigorously tested in the most unlikely places, like geothermal greenhouses buried in the freezing snows of Nebraska, proving its incredible thermodynamic resilience. We analyzed the absolute landscape-altering devastation of the 2022 Florida freeze, where the plummeting temperatures inexplicably spared the ambrosia beetles, wiped out the predatory wasps, and inadvertently unleashed a biblical plague of 95,000 leaf miners upon the surviving, highly vulnerable trees.
We peered through the microscope to examine the incredibly complex systemic warfare of Huanglongbing disease, watching in real time as vulnerable orange trees desperately, tragically choke their own vascular systems with callus in a doomed effort to stop an unstoppable bacterial invader.
And finally, we saw how the ancient, wild genetic keys held by that Australian rainforest shrub are being deployed right now, today, in cutting-edge university laboratories to upgrade the fundamental immune systems of our sweet oranges. We saw how fusing their cells can drop deadly bacterial counts to near zero, utilizing those ancient healing flavonoids to neutralize internal cellular fires, all while this exact same strange fruit simultaneously becomes the hottest, most visually striking, and expensive new cocktail garnish in global luxury dining. It is a stunning, beautifully complete circle of evolutionary biology, human history, cutting-edge genetics, and modern economics.
It is a profound, incredibly hopeful reminder of how deeply interconnected our entire global ecosystem is. But delving into this research leaves me with one final, deeply provocative thought.
What's that?
If the survival of the entire global multi-billion-dollar citrus industry, if the very existence of the simple, everyday glass of orange juice you expect to have with your breakfast relies entirely on the uncultivated, wild, largely ignored genetics of an obscure Australian understory shrub, what other world-saving genetic secrets are currently sitting out there completely ignored in the world's rapidly shrinking wild ecosystems?
Oh, wow.
What undiscovered pharmaceutical cures, what wildly climate-resilient staple crops are out there hiding in the rainforests or the deep oceans, just waiting for a catastrophic global crisis to suddenly make them the most important organisms on Earth?
Wow, that is a heavy, incredibly important, and slightly terrifying question to sit with. If we lose the wild biodiversity of the planet, we aren't just losing pretty landscapes. We are actively destroying the biological backup hard drive for our entire global food supply. Thank you for joining us on this deep dive today. The next time you are standing in your kitchen and you slice open a simple green lime or you peel a sweet orange for a snack, I want you to look a little closer at it. Take a quiet moment to appreciate the massive, unseen scientific, economic, and microscopic agricultural wars that are currently being fought tooth and nail just to keep that simple piece of fruit on your table. Until next time.